Hello, everyone. Welcome to the 2022 New England Regional SACNAS Conference. My name is Carolina Mejia Pena, and I'm the outgoing graduate student president of the Brown SACNAS chapter, and we're so happy and honored to be hosting you this weekend. SACNAS stands for the Society for the Advancement of Chicanos, Hispanics, and Native Americans in Science. Our chapter and others that are represented here are dedicated to fostering the success of Latinx and Native Americans, from college students to professionals in attaining advanced degrees, careers, and positions of leadership in STEM. We work continuously to foster an inclusive community and resilient network of scientists from underrepresented identities in STEM. We strive to promote and achieve true diversity in STEM through professional development, civic engagement, and community building, and we hope that this conference will do justice to that mission. I also want to take a moment to acknowledge that we're lucky enough to be serving this mission in person. Thanks to scientists, health professionals, and our compassion for one another, today we're able to enjoy each other's company and science safely. And if anything, this pandemic has highlighted the importance of scientific exchange and inclusive spread of scientific information, which is exactly what we're here to do this weekend. Before we get everything started, I'd like to take the time to thank the people who made this event possible, because it was no easy feat. First and foremost, I want to thank our chapter members who were responsible for organizing everything from the food that you're going to eat to the keynote we're about to listen to. I've been involved in SACNAS and other student-led organizations at every stage of my education and training, and I've never worked with a group of students more dedicated to each other's well-being and success, so truly, thank you all. Next, I'd like to thank our advisors, Dr. Mark Johnson and Dr. Oluduratimi Adentunji. You support us year-round in more ways than one, and have championed us these past few years to the point where we were given the honor of hosting the regional conference, so thank you. I'd also like to thank Dr. Allison DeLong. Although she is not one of our formal advisors, she certainly spends enough time texting, calling, and emailing with us to be. She was the brains and muscle behind the Nabrit conference, if you were here just a few moments ago, and she helped us so much throughout the planning of our conference, so thank you, Allison. We also thank the Brown staff that have made this conference possible logistically, from IT to facilities, who are making sure that everything runs smoothly, so thank you all. And this is the last one, but I want to thank all of you guys. The conference would literally not happen without all of you, and we're so grateful that you took the time to come to Brown to meet us and share your science and experiences with us. So thank you for making the trek out to Providence, and welcome to the conference.
Hello, everyone, and once again, welcome. And just to highlight the diversity of SACNAS here at Brown and just the community in general, we wanted to say some welcome remarks in some of the languages that represent our community here. So my name is Adriana Coldepeña, and I am a second-year BME PhD student, and I will be saying the remarks in Spanish. So, hola a todos y bienvenidos a la conferencia de SACNAS. Nos alegra mucho que estén aquí con nosotros y esperamos que la aprovechen al máximo. Thank you.
Hello, everyone. I'm Daisy. I'm currently a postdoc in engineering, starting a faculty position in Utah in August, and I would be saying in Portuguese. Olá a todos e bem-vindos à Conferência Regional de SACNAS. Estamos muito felizes que vocês estão aqui e esperamos que aproveitem ao máximo.
Yat shikadosha she Dominic Pablito Yanishia, Tobahanishle, Nast Eja Bashishi, Naslana Doshiche, Nast Eja Doshanele, Kashe Kotadeone Ade, Dons Awake, Ho Dominic Pablito Letchina, Ho Abachideane Tabayataka Awan Chatle, Hello, everybody. My name is Dominique Pablito, and I am a rising third year in the MCB PhD department. I just introduced myself in my native languages, which is Navajo and Zuni. Yeah, my great... Thank you. Yeah, my great-grandparents really instilled in me that the first things that come out of my mouth should be my native language, not English, so that's why I didn't introduce, but I introduced myself first in Navajo and Zuni. So yeah, thank you for coming to the 2022 SACNAS Regional Conference, and today I have the honor of doing the land acknowledgement. Brown University is located in Providence, Rhode Island, on the lands that are within the ancestral homes of the Narragansett Indian tribe. We acknowledge that beginning with colonization and continuing for centuries, the Narragansett Indian tribe has been dispossessed of most of their ancestral lands in Rhode Island by the actions of individuals and institutions. We acknowledge our responsibility to understand and respond to those actions. The Narragansett Indian tribe, whose ancestors stewarded these lands with great care, continue as a sovereign nation today. We commit to working together to honor our past and build the future with truth. With that being said, I'm grateful for the opportunity to share space and introduce Lauren Spears. Lauren is the executive director of the Tamaquag Museum. She aspires to empower Native youth and to educate the public on Native history, culture, environment, and the arts. Lauren shares her cultural knowledge, culture, environment, and the arts. Lauren shares her cultural knowledge and traditional arts learned through her family with the public through the museum programs such as tours, virtual programming, and educational resources. Under Lauren's leadership, the Tamaquag Museum received the Institute of Museum and Library Services National Medal. An educator for 25 years, she has served as an adjunct faculty at Brown University and University of Rhode Island. Lauren founded the Nuitun School, affiliated with the Tamaquag Museum, and was a teacher in Newport Public School District for 12 years. Outside of her work in the museum, she is also an educator, activist, author, and Indigenous artist. Her work as an author includes Dawnland Voices, an anthology of Indigenous writing of New England, Through Our Eyes, an Indigenous view of Mashpinawg Pond, The Pursuit of Happiness, an Indigenous view, From Slaves to Soldiers, the 1st Rhode Island Regiment in the American Revolution, and she co-edited The Key into the Language of America by Roger Williams. Lauren received her undergraduate degree from the University of Rhode Island and her master's degree from the University of New England with a focus on elementary education. Lauren has been the recipient of numerous awards and honors, most notably the 2016 Tom Roberts Prize for Creative Achievement in the Humanities, a Doctor of Human Letters, honoris causa from the University of Rhode Island in 2017. So with that, please help me welcome Lauren Spears.
Askui Kwasin, Natasuis Makasini Pashao at Nahai Gansek, Natasuis Loren Spears at Englishat, Nin Nahai Gansek Nehantek, Kanupiam at Aki at Nahai Gansek. So hello everyone. My traditional name is Makasini Pashao, and most people know me by my more anglicized name, Loren Spears, and I am Narragansett Niantic and an enrolled citizen of the Narragansett Nation. And welcome to our homelands. So I'm going to say quite a bit in my own language. I think it's, as this lovely, wonderful person before me, Dominique, mentioned, it's really important for you to be steeped in the language of the place and the people. And so I will share with you. And as Dominique was mentioning, she was having a little struggles. Our language is very long. The words are very long, so bear with me. Kamian nanan katan taman muank anananaksh kunupiaman anka kakantam batan tam awawan wawan tiawanki kakutam wian tiangan nananag hawan pia hiat nian kamam pak Kawamanan tam taman, paman tam anat wu nuamwana wonk. Wantiawawag kinawan. And what that first passage means, we gather our minds and greet each other and thank the enlightened teachers who have come before us. When we forget to live in harmony, they remind us. And then I'm going to continue from there with my blessing. Katabatash katantua, wuchi wami nawawian nakan wuni tiawank. Katabatash katantua, wuchi wuni kisak. Katabatash katantua, wuchi nipawus ka matakwasag. Katabatash katantua, wuchi kitan. Katabatash katantua, wuchi namas. Katabatash katantua, wuchi wita. Katabatash katantua, wuchi nepeshawag. Katabatash katantua, wuchi peneshamwak. Katabatash katantua, wuchi sokanun. Kacha usha, niempaog ka wayupayanash. Nashbi, Mishatash, and Pianwa, Kachisatau. Kakantamwu, Wami, Sakwan, Kisakwan, Takwanik, Paponi, Pashao, Nashbi, Wuni Tiawank. Katabatash katantua, Wuchi, Wami, Pamantamwe, Tian Tana Gonas Nashin. Katabatash katantua, At Wuchi Tanan Ganonog. Katantam kwamanang at natam mahug ka nashawank. Kanashawanku kachisumai unkunginawan. Kakantam wu ganau aquane wami pishkoni. Nunapi un mawe at wutu anantam nukasaki. Katabatash katantua wuchi wami wunensianat nukasaki. Katabatash katantua namantuanan, wuchi wami wunitiawak ni niwyayanakan. Katabatash wuchi wami kamaguanash namanatum ni nash. And so what I said to you in my language in that blessing is welcome to all my friends and relatives to the homelands of the Narragansett Nation. Thank you, Creator, for all the beauty that surrounds us. Thank you, Creator, for this day. Thank you, Creator, for the sun and the clouds. Thank you for the ocean and the fish. Thank you, Creator, for the forests and the animals and the birds. Thank you, Creator, for the rain, the thunder, the lightning, and the winds. With a storm comes a cleansing. We wish all the seasons, spring, summer, and fall, and winter, to burst forth with beauty. Thank you, Creator, for all living things. Thank you, Creator, for our ancestors, those who suffered mind, body, and spirit. Their spirits wash over us. We wish for peace in the future, and we stand together to care for and protect Mother Earth. We thank the Creator for all the beauty that surrounds us on this earth, that is a gift for all of us to enjoy. We thank the Creator for all our gifts and blessings. Ninaj or Amen. And as I welcome you today and bless this day, I must acknowledge 400 years of conquest, colonization, genocide, land dispossession, enslavement, forced assimilation, detribalization, and erasure of the Narragansett and other indigenous nations of the Americas. I must also acknowledge the bravery, the resilience, the perseverance, and adaptability of our ancestors who ensured our continuation, passing forth our traditional ecological knowledge, our sciences, language, history, and culture, while contributing to the creation of this colony, this state, and this country. There is no U.S. history without Indigenous peoples' history, and there is no Rhode Island history without Narragansett, Niantic, and other Indigenous peoples' histories. This is our homeland since time immemorial. We are still here. May we always remember we are the land, and what we do to the land, we do to ourselves. We must care for Mother Earth to ensure future generations. And it's important, as we all take on the notion of sharing land acknowledgements, that we remember that the land acknowledgement is just the beginning, and that it's really important to follow that by actions. For Tamaquag Museum, where I'm the director, one of the things that we've done as an action is to establish the Indigenous Empowerment Network and to share our traditional ecological knowledge and to represent museum exhibits from a first-person perspective and to partner with organizations like SACNAS and institutions like Brown University to ensure that diversity, equity, access, inclusion, and justice is considered and implemented. Katabatash Akwane Ka Nunantam, thanks, peace, and blessings. And I'm going to follow that blessing and land acknowledgement in my way with a song. This song has been passed down to me from my elders. I had heard it when I was a child, and when I had Nuituan School, we took a lot of Narragansett students to Washington, D.C., to the National Museum of the American Indian, and we took elders with us, and Elasikatau, Firefly Song of Wind, was one of the elders that came with us, and during a ceremony there, she sang this song, and I said to her afterwards, I remember that song from when I was a child. I remember hearing it. Would you teach it to me? And so we sang it together 7 times until I had the song. And that's what we do in our language, in our culture. You repeat it over and over and over again. And the 7 is representing 7 generations. And so she's from a couple of generations before me. And then I'm passing it on to next generations that come. And so this song is Yunaweekan, and it literally means we dwell here. And it understands in those words that our ancestors are here in the soil, our stories are here, our life ways are here, our knowledge is here in this space, and everything we are as a Nahaigansek person is here in this place and in this land. You know how we can, you know how we can, you know we can, we you know we can, oh. We ha, we ha, we oh, we ha, we ha, we ha, we oh, we ha. You know we can, you know we can, we you know we can, ooks. We ha, we ha, we oh, we ha, we ha, we ha, we oh, we ha. You know we can, you know we can, we you know we can, ooks. We ha, we ha, we oh, we ha, we ha, we ha, we oh, we ha. You know we can, you know we can, we you know we can. Once for the past, once for the present, and once for the future. Katabatash, thank you so much.
Thank you, Lauren, for such a beautiful and intentional blessing. My name is Kimberly Mesa, and I am a member of the Brown SACNAS Chapter Executive Board and Regional Meeting Planning Committee. And I'd like to thank again the organizers of the Nabrit and the SACNAS meetings for an incredible program so far, and acknowledge the efforts to amplify the missions of our meetings in a special joint keynote address today. It is my great honor and pleasure to introduce our next speaker, Dr. Christy Redhorse, who graciously accepted our invitation to be here with us tonight and share her work in coronary artery development and regeneration. Dr. Redhorse grew up in Benton, Arkansas, and attributes her love of biological systems to her high school biology teacher, Ms. Parnell's class. This led to her completing an undergraduate degree in microbiology at the University of Arkansas at Fayetteville. Uncertain about next steps career-wise after graduation, which I know I can relate to, Dr. Redhorse took an opportunity to complete a master's degree at San Francisco State University, which she credits this opportunity to the NIH MBRS RISE initiative. This was a pivotal moment in her life for which she thanks Dr. Frank Bayless. It was here that Dr. Redhorse fell in love with scientific research and has not looked back since. Dr. Redhorse is the granddaughter of a Cherokee elder and academic who dedicated his life to the welfare of American Indian families. His research frequently focused on the importance of extended family could have in the welfare of young American Indians, which was intended to influence policies that would keep families together. Dr. Redhorse's lineage and her own personal motivations give her the motivation to provide opportunities to young scientists from many different backgrounds and life histories. Her master's research explored immune cell trafficking at the human maternal interface in Dr. Susan Fisher's lab at the University of California, San Francisco. So enthralled by the biology of pregnancy, she decided to stay in the Fisher lab for her PhD research and explored how vasculature in the placenta incorporates within the uterine arterial network. Her work provided information about how the placenta targets arteries rather than veins, and discovered that the uterus usually lacks a lymphatic vasculature until pregnancy, when uterine lymphatics expand dramatically. She then spent some time at Genentech in the lab of Dr. Napoleon Ferreira, but the work of this lab, of her lab currently, started during a postdoc at Stanford University with Dr. Mark Krasnow. There, Dr. Redhorse identified the progenitors of coronary arteries in mouse hearts. And in her own lab at Stanford, where she is currently faculty, she and her team went on to explore many different aspects of how coronary arteries develop from these progenitors. The Redhorse group also studies how coronary arteries regenerate and respond to injury, and devises methods for increasing their growth to enhance cardiac recovery. For her pioneering work in cardiovascular and stem cell biology, Dr. Redhorse has received awards such as the NIH Pathway to Independence Award, the Cyril Scarles Award, and she was also a New York Stem Cell Foundation Robertson Investigator and received the Judah Folkman Award in Vascular Biology. In 2021, Dr. Redhorse was named a Howard Hughes Investigator. When not working on blood vessels, she's raising her 2 children, 4 chickens, and Labradoodle, named Noodles the Poodles. And so without further ado, let's give a very warm welcome to Dr. Christy Redhorse.
Okay, great. Thank you so much. I am so excited to be here today to talk to you about, it looks like, there we go. To talk to you about the work that my team does, and I also want to talk to you a little bit about my career trajectory and life trajectory. And so, as was introduced, thank you so much for that introduction. I'm so thankful to be here to spend time with you guys today and to be present during that beautiful blessing. That was really a wonderful thing to be a part of. Okay, so in my lab, as was just introduced, we study coronary artery development and regeneration. And I just want to also acknowledge that all the work done in my lab is on the ancestral lands of the Muwekma Ohlone tribe in California. But before I get started with all of the details I want to tell you about a couple of programs to advertise. This is if you're an undergrad thinking of applying to a PhD. My program, the biology program, has this preview program. And if you go to my website, then you can check it out. It's a really cool set of workshops and webinars to help you get some behind-the-scenes tips about the application process. I also want to highlight another brand new program from HHMI called the Freeman Rabowski Scholars Program. And this is going to provide significant funding to early career faculty that are doing amazing science at the same time, going above and beyond to diversify science. Okay, so now I want to tell you a little bit about my life. As was mentioned before, I'm of Cherokee descent, but I grew up in Arkansas, here with my white mother and her family. Here is a picture of us when I was a little girl. And so here's a picture of my great-grandfather, Joseph Red Horse, and he was born in Indian Territory. However, in his young life, he was relocated to California. This was not odd at the time. But when he got to California, he then worked and lived there his entire life, and lived and raised his children among the coastal Indian communities in California, in the Pomo and Maidu and Miwok tribes. And so that's where my grandfather and his brother were raised, among those communities. Here's a picture of my grandfather, John Red Horse. Here's a picture of us now. This was in the 50s, this is now. And as was mentioned, he was also an academic. He was a PhD, and he spent most of his life in Minnesota, at the University of Minnesota, performing research in social work and directing the American Indian Studies program at the University of Minnesota. And so we know, I can go through the life of my great-grandfather, who died before actually the year that I was born, but he was so politically active in the Native American communities in California that I'm able to go through old newspaper clippings to follow his life. But I thankfully know my grandfather very well now, and I want to just highlight one of the tenets or aspects of scientific research that was kind of controversial at the time in the 1980s when he was pushing for it. And his field was social work. But he was really pushing for this idea of when you're a scientist, you need to do research not on communities, but with communities for their benefit. And like I said, this is in the social work realm, but we can all identify with that today because now there's such a controversy in the fact that we can sequence many, many genomes, and we need to really think about who's sequencing them, who's using this data, and who is benefiting from these data. And I really love this consortium here, Native Biodata Consortium. And so they have put together this institute on tribal lands and are really pushing for having natives generating and benefiting from their own data. And so we heard from the blessing about going past the land acknowledgement and actually doing action. And one of the actions that I feel like really felt really good was to do a contribution to this consortium, which I'm not associated with, but that's just one thing, if you're interested in, is available. Okay, so in my own lab, I want to, carry on the baton from my grandfather's work. And so I'm in the position at Stanford I have the resources and the position there to try to, in my own way, influence diversifying science. And so that's what we try to do. Here's a list of some of the students that we've trained that are from underrepresented populations. And then I want to highlight this summer research program, if anyone's interested. I'm a PI on this at Stanford, and it's a chance for undergraduates from different institutions to get a taste of research at a place like Stanford. Okay, so how did I get to California? I followed many, many years later from my other family members, but since I moved from Arkansas to California, I was able to reconnect with them when I was in my young 20s. And so I came from Arkansas after I graduated from college. I went to the University of Arkansas, where my very first research experience was on poultry, in a poultry immunology lab. And one tidbit that I like to say is that I did a little bit of research, and then I decided I wanted to go on, and I applied to PhD programs, but I did not get into any PhD programs when at that step. And now I'm a Stanford professor, so never give up. That's a lesson I always like to say. But just keep trying. But I have to really acknowledge San Francisco State University for having this wonderful Bridges program, which funded me to do a master's. And this was really the time in which I caught the bug for doing research and also had an opportunity to show that I had a knack for doing it. And so that research was in Susan Fisher's lab. She studies the placenta and many aspects of how the placenta develops, and mostly the human placenta. And it was at that time, during my master's, I was able to see the process of writing a paper and publishing papers, and then I ended up never looking back. And so this propelled me to perform my PhD in that same lab at UCSF. Then I did a little bit of time at Genentech, which was mentioned before, in industry, and then finally a postdoc at Stanford with Mark Krasnow, where I started studying the coronary vasculature, and that's what I started my own lab on, and that's what we still study today. Okay, so that is my pathway through science, and also my early information about my life. And so now I'm going to go into the science part and tell you a lot about some of the work that my team has been doing over the past few years. So the major underlying aspect that bolsters all of our research is the fact that ischemic heart disease is the leading cause of death worldwide. And so you can see here, this is in both this is all over the world, and the most common underlying cause of ischemic heart disease is disease of the coronary arteries. And the coronary arteries are these arteries that bring blood flow to the muscle of the heart. And unfortunately, they're quite susceptible to atherosclerosis and other diseases, and this can lead to the blocking of blood flow. And so what we do in the lab is to work under the hypothesis that if you understand how the developing embryo forms the coronary arteries de novo, then perhaps you can go on and identify ways to reform them, to regenerate them in diseased settings such as this. And so I'm going to tell you some work that we've done along this hypothesis, and I want to tell you about a cell type that we're focused on, and that is the endothelial cell. And so the endothelial cell is within the blood vessel. It is the cells that they're a single cell layer that line the lumen. So those are the cells that are interacting with blood flow. And so the reason that we focus so much on endothelial cells in my lab is that they're these highly modal cells. And because they're highly modal, then they are really pioneers of development of the vasculature. They sense and respond to many developmental cues and mechanical signals from blood flow, which shapes the vasculature. And so here's the organ that we study. This is the developing heart. And you can see the motility of these endothelial cells that in an area where you don't have a vasculature, this is a developing organ, then you need to invest it with these endothelial cells, and they do that through this process called sprouting angiogenesis that you're watching right here. And so sprouting angiogenesis will form an immature vessel plexus that will then mature into arteries and capillaries and veins. And during this process, there's the specification and the formation of the arterial blood vessels that we are so interested in regenerating in diseased hearts. Okay, so later on in development, at the later stages after birth, you're going to, or just before birth, then you're going to have this artery and capillary vein network that then expands because, of course, the animal is going to grow in a very large fashion from birth to an adult. And we're very interested as well of the mechanisms that allow this this to expand. And then finally, in the adult, we want to model these cardiac injury processes. And one of those that we use is a myocardial infarction. So this is an experimental myocardial infarction where we block blood flow through the coronary arteries and watch how the arteries naturally regenerate, and then also see how we can take the lessons that we learned from studying these stages to enhance development of these. And so this takes a lot of time, and there are a lot of details, so I'm just going to tell you a couple of stories that we've most recently finished. And so they're going to address the question of how do coronary progenitor sources transition into coronary vessels, and specifically, do they remember their origins? And this is a project by a MSTP student in the lab who recently went back to finish her medical school, Ragini Fansalkar. And then I'll talk about how long these coronary progenitors are competent to make new vessels, and can we enhance this during injury? And this is work performed by a postdoc in the lab, Gaetano D'Amato. So let's start with this first question. And the central theme of this is something that I characterized when I was a postdoc in Mark Krasnow's lab. And that is that sprouting angiogenesis that gives you those first immature blood vessel networks on the heart muscle, those come from two different places. So there's two different progenitors. There's one that's called the sinus venosus, and this migration during this angiogenesis is from the outside into the muscle. And then there's the endocardium, which is on the inside of the heart. This lines the inside of the heart and gives off these sprouts that migrate into the muscle from the inside out. And so here's a description of that. This is what it really looks like. So we've performed lineage tracing, where you label cells and all of their children's cells with a fluorescence, and here it's in green. And here are the cells on an embryonic heart, the coronary vessel cells that come from the sinus venosus, and here are those that come from the endocardium. And you can see that they invest complementary but overlapping parts of the heart muscle during development. And so we were really interested in this fact that there are two progenitor sources for the mature coronary vessels, and we wanted to know why and different aspects of it. And so I'm not going to show you the data because this has been long published, but what we were able to show is that when you have two sources of vessels, you have two places where you can draw upon, so development goes faster. And this is a time in which the cardiac muscle during development is growing very quickly, and it can grow that quickly because it has two sources of vasculature. We also showed that it makes for a more robust system. When you mutate a gene that causes one to be hobbled, one of those sources, then the other one can take over. And so this makes for a robustness to coronary development. But what I want to talk about today is this idea of if you have a structure that comes from two different places, then does that affect the mature cell behavior? So in the adult, can they basically remember their origins? And this is not a detail that only happens in the heart, but we now are coming to appreciate over the later years that this is a common thing that happens in development. We've known it for many, many years, over a century for neural crest cells, but now we're making many of these lines in which we can do a lot of lineage tracing in mice. You may have heard of Cre recombinase technology, where we can knock out genes in specific cells. Well, using these Cres to lineage trace, we now know that this is a very common phenomenon. And this is a review just showing in the heart that this happens in the fibroblast compartment, in the smooth muscle cells that surround the coronary vessels, and in the lymphatic vessels, but it's happening in many other organs as well. And so now that this has been described for many different tissue types at this point, we want to go forward and identify whether having these alternative progenitors, if this really affects the mature cell behavior. And so for us, what this looks like is here. We have this is a tissue section of heart muscle, and it's stained for a marker of blood vessels. And at this time, we think, oh, well, these blood vessels, capillaries here, the small blood vessels, look very homogeneous with the markers that we had for many years. And so we have this idea in our head that it looks like this. However, now, from those lineage tracing experiments that I showed you, we know that they have hidden heterogeneities within them. And so if we use our lineage tracing tools, we can tell that this is not homogeneous, but it's really a population that's come from two different sources. And so how can we determine if these cells remember, if these blood vessel cells remember where they came from? Well, we came up with a few ideas, and I'll show you some data around it. Well, first, you might have this idea that the gene expression, the genes that they express might be different based on where they come from. They might have different developmental capacities, or they might, in the mature animal, respond differently based on what their history was, based on the fact that they came from one place or another. And so first of all, I'm going to show you the data that we got from looking at their transcriptional profile, all the genes that they express. And this was actually an idea that we wanted to explore for a very long time, but we weren't able to really get into it until single-cell RNA sequencing became a technology that was easy to use. And this is because if you use a marker, which is expressed in many, many different cells within the vasculature, then you bring down a bunch of different cells. And so if you're trying to do this high-level comparison, you really need to compare apples to apples instead of having a heterogeneous population all mixed together and try to see if you have differences. And so that's the power of single-cell RNA sequencing. You can take all of these cells and look at their transcriptome as individuals and then pick out who is what subpopulation of the vasculature and directly compare those based on what lineage they came from. And so that's exactly what we did. We took cells from all of these different stages that I introduced earlier, and we have lineage information as well as all the genes that are expressed in each individual cell. And so single-cell RNA sequencing now is so pervasive, I'm just going to assume that most people are familiar with it, but just really quickly, every one of these dots is a different cell, and where it is on these maps tells us a lot about what genes that they're expressing and what state they are in. And so what we do with this type of data is that we understand the cells, what subtypes they are, and what state they're in, what genes they're expressing, by clustering them. And so the idea here for us is that we can see these different clusters, these cells of the vasculature that are in different cell states, and we want to ask whether these different cell states are corresponding to their different lineages, or they came from one progenitor or another, because that would tell us they were remembering. Okay, so now let me first tell you the data from a very early time point. This is 2 days after that sprouting angiogenesis started to happen. And this is the single-cell sequencing plot. And the clustering shows us that we have 3 clusters. One is this pre-artery cluster, which this is one that we had previously identified as how coronary arteries form. Before you even see an arterial vessel, there are these cells that around within this immature plexus will turn on the artery differentiation program, and then those will be the cells that will migrate together and form the arterial vessel. And so we call those pre-artery cells. But for this experiment, we wanted to focus in on the capillaries so that we could have all these different time points, the capillary endothelial cells, to ask this question about remembering their origins. And so the data showed here that there is a heterogeneity to this early capillary network. There's 2 clusters that we see, 2 transcriptional states. And when we looked at the lineage tracing information, we found that this did correspond to the different lineages. The ones that came from the sinus venosus were CAP2 was all within the sinus venosus. And so then Rockney wanted to ask the question, okay, well, it's correlated with lineage in this experiment, but how can we look at the transcription and see if they're actually remembering their origins? And what she did was to identify the list of genes that make the endocardium, these are the progenitors now, that made the endocardium, and the sinus venosus, and asked if CAP1 or CAP2 expressed any of these genes. And so, as you can see here, CAP1 overlaps significantly with the endocardium marker genes, but not CAP2, and then vice versa with the sinus venosus. CAP2 is expressing a lot of SV genes, whereas CAP1 was not. And so what we concluded from this is that at this early stage, 1 to 2 days after sprouting, it did seem like these coronary endothelial cells were retaining progenitor gene expression, or perhaps remembering in some transcriptional manner where they came from. And so this was just a couple days after. This was very early. We were interested in later stages, but this was an important experiment for us because it showed us that our technique could detect this phenomenon. And so now let me show you the later stage. This is right a couple of days before birth. This is E17 in the embryonic day 17 in the mouse. And again, we have 2 capillary states that the endothelial cells are in this developing heart. Now this is a much more mature. You can see here in the schematic, there's arteries and veins, but let's focus here on these capillaries. And what we found was that they did not, at this point, segregate based on their lineage. And when Ragini overlapped the marker genes, she found that there was not that pattern of overlapping between the two. And so what we concluded here was that the cell state is not related to the lineage at this later time point. So they had, in essence, transcriptionally forgotten where they came from. But there are these 2 capillary clusters. There's a heterogeneity to the capillary network in the developing heart at this point, and so we wanted to know what was behind these different cell states. And one of the ideas that we had was that perhaps it was location. And so I'm going to show you one of the marker genes that we use to ask the question, is the difference in their transcriptome related to that? And that's CAR4. This is a gene that's involved in carbon dioxide processing. And so this is a slice through the heart right here. And as you can see, this CAR4 is expressed on the outer wall of the heart, or we call these the free walls of the heart, but not in this inner portion of the muscle, the septum. And so this and other marker genes that we looked at told us that CAP2 was in the free walls of the heart, the muscle on the outside, and CAP1 was the endothelial cells within the septum. And so later on, it's not the source that causes heterogeneity, it is the location. And so just to remind you, since many of you are probably not 100% familiar with the different structures of the heart, this is myocardium here, cardiac muscle, in different areas and different chambers. And so again, we have these markers that we stain and the cells look all the same, but in the middle, we see that in the septum here, as opposed to the free walls, we have a different transcriptional profile of the endothelial cells. Okay, so now can we take those genes that are expressed differently among those 2 compartments and try to get some data about why they're different? What is their function? Well, we went through all the genes that were in one capillary population and the other, and what we found was that there was the concentration of the expression in CAP2 of genes that in previous experiments have been shown to be induced by blood flow. So blood flow is a mechanical force that when it imparts its force on the endothelial cells, it causes changes in transcription. And so we know that KLF2, KLF4, and others are upregulated by that. And so conversely, in CAP1, we found that genes that were known to be induced by hypoxia or low oxygen were induced. And so these are genes that in low oxygen environments are induced by the HIF1 alpha transcription factors. And so that kind of makes sense because if you don't have flow, you're going to have lower oxygen. So that all fit with this hypothesis that we're using to go forward, that blood flow in the septum during development is later to become established and compared with the free walls. Okay, so since we found that in this later stage in development, they didn't transcriptionally remember their origins, we didn't really expect them to re-remember them in the adult, and that was the case. So here's the arteries and the capillaries and the veins in the adult. And what you can see here is that most of the capillaries go into this state called the CAP2 state. And so that's good because you wouldn't want part of your cardiac muscle to be in this hypoxic low flow state. So that makes a lot of sense. So conclusion is most endothelial cells convert to a similar state by adulthood and do not have transcriptional remnants of their unique lineage. And so what's happening during development here to put this all together into a model is that there's two primary sources for coronary vasculature, the sinus venosus and the endocardium. These move on to the heart to invest the heart muscle for a little while. They continue to express indications of their progenitors, but later on, they downregulate those progenitor genes. There is heterogeneity because some parts of the heart are slower to receive blood flow, but then in the adult, there's convergence into a one cell state for the most part of all of the endothelial cells. And so this led us to scratch this out on the board of possibly remembering their sources. But that's just one aspect. Gene expression is just one aspect. What about some more functional ways of looking at this? Well, one is developmental capacity. And we had already done some experiments to address that in earlier work. And so my postdoc, Bikram Sharma, had shown that whenever you inhibit sinus venosus sprouting, then the heart has some abnormalities for a little while. But if you give it time, the endocardial-derived vessels will grow and compensate to make up for the loss, and heart function after a while will be normal. And so developmental capacity doesn't seem to be different. How about response to injury? Well, this is one of the last experiments that Ragini performed. And what she did was to lineage trace one of these sources from the endocardium, and then wait until adult stages and do one of these injury experiments that I described, this ligation of the coronary artery to stop blood flow manually. And so what she did was then to use an assessment of endothelial cells being activated by the injury and seeing if they were better at being activated, whether they came from one source or the other. And so labeling the endocardium, doing the injury, and labeling with EDU. And what she found was that similar to the transcriptional analysis, there was not a difference, statistical difference based on which lineage the endothelial cells came from, with driving their response in this way to the injury. However, she did have some indication, and also Cariala Talo's lab published a paper that supported this notion in that, again, location might be driving differences, not lineage, but location. So in the inner wall, there was a different proliferation, whereas if the endothelial cells are in the outer wall, they proliferate more. And this has been repeated in other groups, as I said. So then we scratched this off of the list and pretty much convinced ourselves that endothelial cells during development are extremely plastic and can take on the shape of whatever tissue that they migrate into and invest. And this is a real plus for regenerative medicine, we think, that we can really push these cells for a while at least into what we would like them to be. Okay, so now I'm going to talk about the second part, which also involves these progenitors undergoing sprouting angiogenesis and investing the heart muscle. But this is the idea, addressing the idea of when do these progenitors spit out these sprouts, and for how long can they continue to do that? because that's also important from a regenerative medicine standpoint, because it would be really great if in the adult we could get this to happen. Okay, so here I just showed you that the two sources will converge in the adults, and when do the progenitors emerge? And we came up with two pretty easy to come up with ideas and models, is that there could be a continuous or a restricted model. And so the continuous model we think of as kind of like the treadmill model, where you, during this developmental process, you have your progenitors, and they're just constantly giving off little sprouts of endothelial cells as the heart is growing into a larger size. The restricted model, I think of more as like a cannon shooting off, where there's at one point in development where you're going to have this conversion from progenitor to coronary blood vessel, but then at the time points in which you need to expand the heart, then you have new vessel growth that is only from expansion of the pre-existing vasculature that initially developed way back when in the cannon shoot-off time point. And so the way that we address this, the way that Gaetano went about this, was to do some lineage tracing experiments where he would lineage label the endocardium, one of the progenitors, at different time points in development. And then he would ask which of these pulses then labels the coronary blood vessels that come from them. And then that would give you the answer of whether they had the capacity at this stage to give you coronary blood vessels. And so more specifically, what this experiment looks like is that we have a mouse that expresses Cre, which is tamoxifen inducible. You have to inject tamoxifen to make this Cre line, which is expressed only in the endocardium. And when you activate it by injecting tamoxifen, then that's going to make a fluorescent marker in the cells that are in the endocardium and all the cells that are born from them. And so he gave the tamoxifen to do these pulse labels at different time points in development, different embryonic days, and then analyzed them late in development. And I'm going to show you the pictures that he did from this experiment that come from this area. So this is the area that gets their coronary vessels from the endocardium. So we're going to look at that in this experiment. And so here's the front side of the heart, or the ventral side, from these experiments with these different labelings. And you can see here, if you look for that lineage trace, it says TD tomato, so it's really red, but we're showing it here in white, that there are a lot of white blood vessels here, a lot of white capillaries, which go down in their numbers as we go towards this embryonic day 13.5. And that's quantified here. So the number of cells that come from the endocardium with these different labels go down across time. And what I want to tell you here about these specific dates are that this goes down before the sprouting even occurs in these areas of the heart. And so what happens is there is a transition from endocardium to coronary blood vessel very early, before you even see the sprouts. And then after that happens, they invest the heart and really expand, and then grow, and do not come from the progenitor after this very early time point, for the most part. And so that told us that this was the right model, this cannon model. And I have this triangle here because I didn't show you all the details of the data, but if we do this experiment that I described, we do find the cells will trickle out at these stages. Most of it happens here before sprouting, but we can get, after birth for a few days, we can get some cells transitioning. And so that bodes well for us trying to maybe push this system and enhance this little trickling that they do have in order to, in an injury setting, perhaps revascularize the heart. Okay, so one idea that Gaetano had was to use this fact that the progenitors only give you coronary blood vessels early and not late, use this as a way to shorten his list of candidates from single-cell RNA sequencing data to identify the genes that were important to drive this angiogenesis. And so this is the data, the same data that you saw earlier, which has lineage tracing and single-cell RNA sequencing from these 2 time points. This is when the sprouting is happening, and this is after we don't see much more lineage tracing. And so if you look at the full data set, I showed you a smaller portion of the data set to hone in on those capillaries earlier, but really that data set has a lot of cells. And so if you look at it here, what you can see is that both we have a lot of cells and that we can isolate those cells that have the lineage mark. So this is lineage tracing. This is the blue is the expression of the lineage trace marker. So we can see these are the cells, these are the coronary cells that come from the endocardium, and this is definitive because we have this lineage trace. And so the first thing that I want to show you here is that at E12, there's this bridge between the coronary vessels and the endocardium. And so this bridge is composed of cells that have half endocardium and half coronary vessel transcription. So they're transitioning from one cell type to the other. And we don't see this bridge at E17. And so this recapitulates what we saw in the lineage tracing data in that there's no longer a massive differentiation from endocardium to coronary vessels at this time point. And we know that this happens because we can then analyze the data set that has the endocardial lineage tracing within it. And so we know we labeled here, we're tracing into here, and so we know that the trajectory goes this way. And this is the trajectory, again, that we don't see in our later data set. Okay, so now we have these 2 data sets where we can identify the cells that are transitioning from the progenitor endocardium to the coronary vessel. Now, what are the genes that are marking that area, that transition, that might be important for the actual activity? Well, Gaetano went through a lot of different genes, and one that really stuck out in his head that was really specific was BMP2. And we talked about BMPs earlier today. They are a secreted molecule that cells secrete and use to signal to neighboring cells. They bind BMP receptors. And so when we used cell chat to look at the gene expression of the BMPs and the BMP receptors, then we can make a hypothesis about the direction of signaling. And what we found was that the BMP2 is being expressed in these transitioning cells, and the receptor is expressed primarily in the endocardium. So we made the hypothesis that these transitioning cells that are differentiating are going to have a feed-forward loop to signal back to the endocardium once they're coronary to amplify this process. And if that was true during development, that they're amplifying differentiation, then Gaetano reasoned that if he, in an explant culture of coronary vessel sprouting angiogenesis, if he added BMP2, he might be able to enhance that process. And that's what he did. We have this assay to where he can lineage label the endocardial cells, and then dissect out the heart, and in culture it in a way in a media that will be allowing for coronary vessels to sprout out. And so this media contains VEGF-A, which he either did or did not add BMP2. And so here is an example. This is where the sprouts should be happening. This is the vehicle. This is just BMP2, and BMP2 didn't do anything on its own. So in vivo, it probably has to couple with another protein. Here's the media that is known to induce sprouts in this assay. It contains VEGF-A, vascular endothelial growth factor A. And then if we add BMP2 to VEGF receptor A, then we enhance this process. And so from this, we concluded that BMP2 does stimulate sprouting of the coronary vessels from the endocardium. And so finally, he wanted to ask whether he could identify, when he identified this activity, then could he use that knowledge then to go back and stimulate angiogenesis in an injury model? And what he did was to perform this coronary artery ligation, again, blocks the blood flow and stimulates the production of an infarct. And then he added a AAV virus that either expresses the control protein or BMP2 and looked at angiogenesis from the endocardium. And as you can see here, this lineage trace is showing that there is some angiogenesis in this region after the injury, but it is greatly enhanced when we infect the myocardium with the BMP2 expressing virus, which is here. So we conclude that we have used the developing heart to identify molecules in which we can enhance angiogenesis in injured hearts. And so to summarize, what I've told you is that, one, we study coronary development and regeneration across different timescales in a mouse life. And there is this phenomenon of two progenitors that give rise to the coronary vessels in the mammalian heart. And we know from single-cell sequencing that this is also true in the human heart as well. But these two sources converge to the same state. And I didn't show you the data that mouse and human are highly similar, but the endocardial differentiation largely precedes the sprouting event, and this is guided by VEGF and BMP2 expression in that area of the heart. And we use this information that BMP2 is driving this process in the embryonic heart to enhance it in the injured heart in postnatal injury models. And so finally, I want to thank you all for listening, but mostly thank my lab here. Most of the work was performed by Gaetano and Ragini. Ragini has since left the lab. This was just the other day in our sunny California spot where we like to go and have lunch, and we go and have lunch outside. But this is Ragini because she was not there that day. She's gone back to medical school. And finally, to thank my funding sources and our collaborators on this project, especially Alessandra Vigilante at University of College London, who did the informatics or helped us with the informatics. And I'll take questions.
If anyone has questions, just raise your hand and I'll come bring you the mic.
Hi, thank you for your talk. It was pretty interesting. Is there any data available on single-cell spatial, like 3D of the heart that we can use to map, like, because I think it's really interesting, this thing about positioning. So maybe even if you didn't have the data, but there is some data available that you can use to kind of see different signatures.
Right. Yeah, so around the time that we were publishing this, a spatial transcriptomics paper came out alongside of it, not in the same journal, but around the same time. And so we tried to use that data, but it wasn't high enough resolution. But we have plans to perform that in our own lab. But yeah, it's a great idea because we want to do it for the mouse, but especially for the human, because we have all the data that you see here, we have overlaid with a human. She also did the human situation, but then we want to do the spatial transcriptomics on slices of the human heart because you can't do the lineage experiments. So you really want to get a good idea with that system.
Hi, that was really beautiful. So you mentioned that you looked at the single-cell transcriptomic states, and but you also mentioned about the chromatin states. So have you looked at that, whether there's predisposition to responding to signals, or where, like, if they're responding to location cues from other cell types, maybe cardiomyocytes or other?
Right, yeah. So we did all of this before the multiome, but then since then, we've performed multiome, but for a different project without the lineage tracing. And we're identifying, different locations among other species because we're using it to compare other species to see, because the location is so important, how they affect epigenetics. yeah. But yeah, it's a really good idea.
Oh, CO. So have you considered looking at situs inversus mice?
Sorry, what?
Situs inversus?
Oh, yeah. So we considered looking into that when I first started my lab, and we were studying how the ostia attach, so how the coronaries attach to the aorta so they can receive blood flow, because that's super important, the positioning. But we haven't since then looked into that.
Thank you so much for your talk. This was I learned so much, so thank you for that. So I have a more technical question. So I know the LID is known as the widowmaker, and in this experiment you were ligating the LID. Is the anatomy different, or are you ligating it in such a way you're inducing like a slow kind of like onset of ischemia in these mice?
Right, no, it's an acute. So it's modeling an acute MI where you have flowing blood. In the situation in a human, it would be analogous to getting a clot, and then a clot getting lodged in the LAD, which happens a lot. And so it's not that slow process, but we're trying to now develop a mouse model where we can genetically change the cells so that we can get that slower process.
Thank you.
Yeah, the anatomy of, and that, so this experiment has some variability because the anatomy from mouse to mouse of the mouse's LAD, it's actually not anterior descending, it's actually on the side more, which Pam will show amazing pictures of tomorrow if you guys are here tomorrow. But yeah, there is some variability because of some technical variability, because of anatomical variability.
Oh. Hi, thank you so much for your talk. It was super interesting. I just had a question. So I work with bats here and one of the Brown labs, and I was just wondering, like, would you be interested in repeating this experiment like with another species of animal in the future, or like replicating it?
Right, yeah. So one of the things that we want to do, and at one point we were trying from Berkeley to get some bat hearts. So we would love to Pam is going to show again these amazing images of the anatomy, and the anatomy is different from different species. And so using that natural variation to learn more about how they develop is what we would like to do in the lab and what we're already doing. But we're using the anatomy of the guinea pig versus the mouse, because the guinea pig has these collateral arteries, natural, and collateral arteries are like natural bypasses. They're two connections between arteries. And the guinea pig makes these that are large enough to completely reroute blood flow in the case of an occlusion. In the case of mouse, they don't have that, and so that's why they have heart failure when you do that experiment.
Thank you.
Yes, but please talk to Pam about the bats and imaging.
Yes, that is very cool. Also very cool talk. And I was just wondering, mice and humans can develop cardiomegaly, and it can be, early onset or late onset, and the heart just becomes huge. Is cardiomegaly accompanied by commensurate increases in sprouting angiogenesis?
Yeah, it is, because the metabolism of the heart. Well, in certain cases it can and it can't. Like in certain diseases, they won't be co-developing, and so then you'll have certain problems because of that. And in certain cases, and they've also shown experimentally that if you increase angiogenesis, that will affect the cardiomyocytes and make them become larger and grow the heart just because you have more blood vessels. And we showed we didn't show in that model, but we showed that during development, the blood vessels themselves secrete things that tell the cardiomyocytes to proliferate. And so it can go both ways. The heart grows and expands too much from a cardiomyopathy, and then angiogenesis will ensue. And then also if you unnaturally overexpress angiogenesis, then that will make the heart also grow abnormally large.
Wonderful talk. I was just wondering about the cells that were in that bridge area in your single-cell RNA sequencing, and whether or not there's anything different about them that you've identified, such as differences in the metabolism.
Right, yeah, so there are some differences. We honed in on BMP2 for the first pass, but there are some others. And I don't think metabolism came up as like a go term in the differentially expressed genes there, but there are some others.
Hello, I'm Miles Bartholomew. That was really awesome because I like single cell stuff. I have a question about, like, I guess this is, like in tumors, tumors often have their own microenvironments and do angiogenesis. Do you think any of the mechanisms that you've observed here are conserved in that model, or?
Right, yeah, so there are data sets out there of tumors and their associated endothelial cells, but I haven't really followed what they're saying about the vasculature. I do know that the vasculature is like incredibly unstable because there's such high growth rates in the tumors that you have hypoxia, and that drives permeability of the endothelial cells. And then another issue is that those mechanisms drive angiogenesis of capillaries, but not maturation of the arteries and veins. So you get this weird vasculature that's just not that good. It's overpermeable, so it's leaky, and it just has a bunch of small vessels. And blood flow can't be driven very well if you don't have the proper hierarchy. But on a single cell level, yeah, I'd have to go through that literature to see.
Any other questions?
Is the hypoxia in the early stage development, does that have a developmental role, or is it just a maladaptive developmental accident?
Yeah, that's a really good question. I feel like everything has a role if it happens by this point in evolution. And so it would be really fun to test what would be the effect if you made it not hypoxic. Would that like stunt some kind of growth because it feels like it's already at this end stage, or what? But yeah, that'd be a fun experiment.
Okay, I think we're actually out of time, so let's give it up again for Dr. Red Horse. So we'll just have some brief remarks about tomorrow's schedule, so just sit tight for a little bit, and then there will be a reception following.
Thank you for that wonderful talk. So before Dr. DeLong gives the closing remarks, I just want to go quickly over the schedule for tomorrow for those who will be joining us. So most of you should have access to these, whether it's physically or through our Twitter, website. You should all have access to our schedule. But basically, the plan for tomorrow is to start another round of check-in breakfast at around 8 a.m., and then we'll have a welcome and orientation. We'll have some addresses by some Brown faculty, and then we will have the concurrent oral presentations. So if you look at the schedule, we actually have them divided by disciplines and topics. So you could go from one to the other, but you would not be able to attend the ones in the same slot at different rooms. After the session with the talks, we will have a keynote by Dr. Omolola Inyola Adefeso, and then launch and the opening of the grad school and career fair. Something you should take into account is that the career fair, grad school and career fair, will actually be open through several of the other events, but there is still enough overlap so that, for example, you wouldn't have to miss the poster symposium and you would still be able to attend some of the other concurrent professional development sessions, which are also detailed in the schedule. So if you go to the last page of the schedule, you should be able to see in detail all of the professional development sessions we will be hosting tomorrow. And if you have any questions, please feel free to flag down any of the organizing committees. We all have these little beige flags, so feel free to ask us any questions. And once again, thank you all for joining us today. Now, we have some closing remarks from Dr. Long.
So thanks to Adriana. Whoops, and yeah, I will. Apologies. So I want to thank Adriana, and I'd also like to thank Dr. Christy Redhorse for a fantastic joint session keynote talk. For those of you who were fortunate enough to be here for the early events of today's Nabrit conference, I really want to thank all of the speakers and panelists for the fantastic job that they did. It was really a wonderful day of events, and I think all of us really learned a lot and have a lot of ideas and thoughts to take home and think about. I'm very excited to announce that we are already planning next year's Nabrit conference, which will be our 5th Nabrit conference, June 8th and 9th. And all of you are welcome. And I will start, or before I go further, I want to thank all of the sponsors of this year's Nabrit conference. I want to thank the audience members as well for great discussions that we have had during both days of the meeting. All of the members of the organizing committee, starting with Kimmy and Carolina and Miles Mundy and Mark Johnson, who's also been recognized as a SACNAS chapter advisor, Dr. Phyllis Dennery, and Michelle Dawson. I want to give a really special shout out to all of the SACNAS members who have, in many cases, worked every year in organizing the Nabrit conference. It's really been a privilege to work with them. And a special shout out to Amanda Ruiz, who isn't here today. And even after she learned that she wouldn't be able to attend the Nabrit conference, continued to work tirelessly and really at a very high level to help organize the meeting. So she really deserves special recognition. So with that, thank you all. And so now, next year's Nabrit conference, we're very excited to announce that we have 2 keynote speakers already lined up. And so we will have keynote addresses from Dr. Cassandra Extavor from Harvard and Dr. Alejandro Sanchez Alvarado from the Stowers Institute. So thank you for joining us, and we hope to see you all next year, June 8th and 9th, here at Brown in person again, we hope.